We started the first lecture on Tuesday, kind of doing a motivation review on the kind of biological problems that could benefit from insights from physics-based models. And so in order to build on those things, where we ended off last time was, we said, well, we're going to take a cell, in this case, single cell E. coli, and we're going to use this like our hydrogen atom. We want to sort of dissect it, get a sense of length scales, energy scales, and time scales. That would be of interest for not only a physicist, but also for a biologist. And so the type of questions that we're interested in answering are, if I know the volume, if I know the mass of the E. coli, if I know the relative percentage weight of dry weight versus wet weight, so water, how many proteins, how many water molecules, how many ribosomes am I fitting into a cell that's about 1 micrometer cube? So if I can answer that question, then I can also answer another question, which is, what's the concentration of particles inside the cell? If I know what the concentration of particles is, I can also answer, what is the typical distance between the particles as they diffuse around? And if I know their typical distances, then I can also ask, well, if I imagine a particle undergoing a Brown and random walk, or let's say undergoing a motion, is it fair to describe it? Can it be described as a standard diffusion, or is there some other type of anomalous behavior going on in the dynamics of the particle itself? So where we're going to go is that the cellular environment is very crowded. It's a crowded environment with lots of neighbors around, and so we need to understand what the nature of that crowding is, what causes the crowding, what are the timescales associated with the elements that make up the crowded environment. And what we're also going to do today is try to understand, why is it that biology or biological systems work so efficiently? And if we want to understand that, we need to break it down in terms of energy usage, energy transformation, energy fluctuations. And so that's what we're going to try to do today. So this is where we were. These are just different images of E. coli. And that's the type of dimensions that you want to keep in your mind. Okay, roughly 2 micrometer by 1 micrometer. And what I would hope that you would take away, for example, by the end of the class, at the end of this course, is being able to answer very simple questions like, if I have a particle undergoing diffusion in 1D or 2D or 3D, and it was with a specific mass in a specific environment with a different viscosity, what would be the typical time scales it would take for the particle to diffuse from one side to the other? Okay, so those are the types of things that we wanted to try and get a sense of. Okay, so just to calibrate you on the numbers again, to remind you, so the volume of the bacteria, roughly about 1 microcubed. The mass is picograms, 10 to the minus 12. Okay? The typical radius of a protein, so these proteins, and you will see these come up often. They have very complex morphologies, but what we're trying to get a sense of is, what are these typical lengths? So if this is the center of the protein, how large is it? And so the typical radius of a protein made up of several hundred amino acids is about 20 angstroms. Okay, that's the length scale. And what we're going to see, and this is really the heart of why biological systems work very efficiently, is that we're dealing with force fluctuations on the order of piconewtons. And where this comes from is thermal energy at room temperature divided by a length scale. That length scale is nanometer or angstrom. And it's only on this length scale where you get piconewton sort of fluctuations. And this is an important point that we're going to address in more detail. Please.
What is DA?
DA is Dalton. Dalton is the name of a chemist, and it's just a unit of mass. So 1 Dalton is 1.6 times 10 to the power minus 24 grams. I guess it's just a way to ease representation of very lightweight biological things. Okay, let's review. Fine. So the first thing we want to ask is, I have a cell, I have a very crowded environment. Okay, so you probably saw these types of pictures when you were taking your biology course. So that's your cell. This thing here is the cell membrane. Okay, there's nucleus, there's DNA, there's water, and there's lots of proteins. Okay, so this is 1 micrometer cube, the volume, 1 picogram. 30% of the weight of the protein of the cell is dry weight. 70% is water. Okay, so we want to know, on average, how many proteins are there in a cell? Okay, so you already answered that for us, and you said?
No, I think that was for water.
No, wait, no, really?
Yeah, 10 to the power 10 water.
10 to the power 10, okay. I thought you also answered, remember you said 10 to the 6 proteins.
No, you said it.
Oh, did I? Okay. Okay.
So you said exploding.
Okay. Right. So, but now I can't play the guessing game, because I was hoping I could play the guessing game and ask. So okay. How would you come up with a number for this? How would you estimate the number of proteins in the cell, given this information? So, I've told you 30% of the cell is dry weight. That includes protein, it includes fats, DNA, etc., etc., etc. Okay. I'm telling you that your typical protein maybe has about 300 amino acids. Now, with that information, how would I just get a sense of how many proteins there are in the cell? Yes, please.
I think since we know that we have 10 to the 10 water.
So, okay. don't start from there. Yeah, sure. This would have been a nice way to do it. Because you say you take 30% of that.
Exactly.
Okay, but that, yeah, that, so then we'll ask how you get 10 to the 10. Okay? But let's start with, let's say I didn't know. I don't want to use that value first. How would I, so let's say, how about, so the first thing that I would do, which is not correct, is I know what the volume of the E. coli is. I can estimate with some radii what the volume of a sphere is. Well, estimate what the volume of a protein is, assuming that it's a sphere. And then I say, how many proteins can I pack into that volume? How many spherical-like proteins can I pack? Okay? That would give me, would that be correct? Is that okay to do?
But we should know the volume of protein, one protein that.
Okay, let's say I know that. So I say it's 4 over 3 pi r cubed, where r is 20.
No, because it's not a closed packed structure. We know where the volume when it's
Yeah.
full.
Yes.
When it's dry, we don't know the volume.
Exactly. So this would overestimate the concentration of proteins in the cell. Okay, because it assumes that everything is packed very nicely at insanely high concentrations. Okay, so, and it doesn't, so more physically, it doesn't include excluded volume. Okay, so every protein not only comes with the protein, but it comes with its excluded volume, which is very analogous to the repulsive wall of, the Leonard-Jones potential. It's the same concept, except the sigma is much larger. Okay, so that, you could do that and you get sort of an upper bound to the number of proteins in the cell. But, so if I know this, I know the total mass of all the proteins in the cell. If I assume that your typical protein has 300 amino acids, the total mass of each protein is 30,000 Daltons, which you can convert into some unit of grams. Okay? And I know that 30% of the...
Oh, that's the total mass of each protein?
Which one?
30,000.
Yeah. It's just a guess. Okay? So with this, you can answer the question. So, 1 Dalton is this guy here. Okay? So the total mass of protein is this object here. The mass per protein is 30,000 times this. And so the total number of proteins is on the order of 10 to the 6.
I thought that 30,000 was the total mass of protein in one cell.
No, per protein. Per protein. Okay, so that's, okay, when you do the numbers, it makes sense, but it's a lot of protein. Because now what I want you to start thinking about is, if I have all these proteins packed on a lattice that I would imagine on the cell, what would be the typical distances between two protein molecules? And it's not a very large number. It's something on the order of tens to 100 angstroms. So 10 nanometers, which is not huge. Okay, so, and this is going to be sort of where we're going into, which is that what makes a biological system special is going to the length scales of nanometers, which is why also, as you know, there's a lot of hype on using nanotechnology for material science, for catalysis, for all these other, and we're going to try and address that question. But this is something that you should really understand deeply, is that we're going to see that going to the nanoscale is what makes energy conversion very, very efficient in biological systems. It doesn't happen at any other length scale like it does at the nanoscale. Okay, so we'll dissect that a bit better later. So that's protein. So there's a lot of proteins in the cell. How many water molecules? 10 to the 10. How did we get that number?
Okay, so-
Wait. Wait. So I know 70% of 1 picogram is the total mass of water. Right? True. Do you accept that statement or not? I know that 70% of 1 picogram is the total mass of water in the cell. Yeah, the mass of the cell is 1 picogram. 70% of it is water. So 70% of 1 picogram is the total mass of water. Okay, the molar mass, yes, and then molar mass is 18 grams per mole. You plug in 1 mole is Avogadro's constant, boom, and you get the total number of waters in the cell. Okay, so that's this. Okay, 70% molar mass Avogadro. So on the order of 10 to the 10 water molecules. So this is actually, I want you to get comfortable with these types of very simple estimates. I'll also give you some homework on this, but this is the type of thing that one of the things you'll be tested on in the exam. Okay? Okay. So there's a lot of water in the cell. So, when you see pictures of cells, you've got to imagine a lot of solutes surrounded by a big bath, and the spacing between the solutes, as we will see, is on the order of 10 nanometers. And what's separating or sandwiched in between there is a lot of water. Okay. So we can go through many other sort of calculations like this. Ions. So there are also ions in the cell. The ion gradients, is what determines things like the osmotic pressure that cells feel in different conditions. Okay, so the typical concentration of, let's say, potassium ions in the cell is about 100 millimolar. Okay? So, well, you could also estimate how many potassium ions there are in the cell using a very similar logic as we did for the molar mass of water. I'll just give you the answer for this. So, you have about 10 to the 7 potassium ions. Again, 10 to the 7 potassium ions, 10 to the 6 proteins, 10 to the 10 water molecules. So, and we're just getting started. There's many other things that are in this crowded environment. Okay, one of the things that I want you to do for your homework is the following. So, I want you to estimate how many ribosomes there are in the cell. So ribosomes are the machinery that generates proteins. and 20% of the cell protein is ribosomal protein. So that's one fact that you need to know. The mass of one ribosome is 2.5 megadaltons, okay? And 1/3 of mass of that is protein. Okay, so not all the ribosome is made of protein. The ribosome is made of many different things. 1/3 of that 2.5 megadalton is protein. So I want you to try and get a guess of an estimate of the number of ribosomes that will be in the cell. Okay. We could continue doing this exercise, put DNA, but I hope you already have a sense that this is a very crowded environment. Okay, lots of different things talking to each other. So this is the picture that people often show of the cell. Okay, so the cell is not an empty space with a few things sitting everywhere. You've got all these different things. Okay, and so and what we're going to spend a lot of time doing now is dissecting various length scales and energy and time scales associated with not all these processes, not all these objects in the cell, but some of them. Okay. So I already mentioned this to you. A concentrated, crowded environment has implications on the concentration of particles in the cell. And that has implication on the typical distances between the particles in the cell. And so the rule of thumb is the following. So 2 nanomolars roughly corresponds to 1 particle, 1 protein in a volume corresponding to a single bacterial cell. And so this is the log-log plot going number of molecules versus concentration. So this is obvious. Okay, I go to high concentration, lots of molecules. Now if I put these molecules on the lattice, you can see that as I increase the concentration, the typical distances between the particles drop. And so I have how many proteins in the cell? 10 to the 6. So this is up here somewhere. It's down here in concentration. So what this is saying is that the average spacing between particles in the cells is quite small. So why does this matter if you're coming from let's say, from the point of view of a physicist? Well, if you're trying to understand the physical processes of biological systems, there are going to be lots of strong correlations between the particles, because they talk to each other a lot. And so one has to worry about, for example, building models where you only have a single particle in the cell versus something that is more realistic and you have multiple particles in the cell. Okay, because these distances are not very large. Now, that's let's say more of a from the point of view of a model. The more practical implication is the following. So often in whatever types of numerical work or analytical work one does, you always want to compare with an experiment. Okay, because the experimentalist measures something. And the experimentalist can measure something in 2 scenarios, either in vivo or in vitro. In vivo is when they actually do the experiment in the real cell in its cellular conditions. So typically they use mice to do this type of stuff. Okay? That, if I do a measurement there, I'm accounting for all the crowding that's going on. Another way to do the experiment is to do it in vitro, so I do it in the lab. So one has to be very careful when one makes a measurement of a biological process, in this case and in this case, and you want to understand it, using a simple model, one has to remember that, of the consequences of this effect. Okay? Are there any questions before I move on? It's all clear. So, okay, so now we sort of have we're building a picture of number of entities in the cell, number of particles that we have to think about. Of course, these things are not static. They're dynamically evolving things because of fluctuations. Okay, so the next thing we need to do now is to build our intuition on the relevant time scales in biological processes. Okay, so what's your feeling? What do you think are the relevant time scales in biology? Is that even a meaningful question? When you think of time scales, what comes to your mind? So, okay, I know of one time scale, that's the age of your life. Okay, so that's a trivial one. That's probably that is the longest time scale of your of a biological process. Okay, so your lifetime. Okay, what if we go back to our first lecture where I talked about all those different examples?
So the other one is when newborn gets ready to reproduce. So
Okay, so that's 10. No, well, gestation. No, no. That's the time scale of how long the daughter or son lives before it's released. So you're talking about
let's say
post.
yeah.
Yeah. So that we're still talking years, months. Okay. Let's zoom down more.
Like we are only focusing on humans, so I think we should go on cells right now.
but humans are cells.
No, like one single unicellular.
Okay. Okay, that's fine. Let's talk about the timescales of this object. What?
So you said that for Ebola, I think it's 3,000 seconds.
Mm-hmm.
So to double it up means so we have one dynamics.
Yes.
The total the rate of creating, let's say, some amount of protein or some amount of water is this time.
Mm-hmm. Okay, very good.
Unique number of proteins.
Excellent. Okay, so that's 3,000 seconds is the time it takes to generate a daughter cell. To take one and split it in two. Okay, and now this is a very good point because we just said there are 10 to the 6 proteins, 10 to the 10 water molecules. So on this time scale, I need to go from this to this. Both 1 and 2 roughly have to have the same number of proteins, same number of water molecules. So I have to generate lots of proteins per second to create the 2 cells, and I have to also suck up lots of water per second to create a cell. Okay, good. So that's still seconds. We're still on the second time scale. Let's zoom in more. Let's zoom in more to the length scale of atoms and molecules.
We can just divide this by 10 to the power 6. 3,000 by 10 to the power 6.
Sure.
So that would be per protein.
That's right. Well, no, that's the number proteins generated per second.
No, that's the number of proteins. Yes.
Number of proteins, 10 to the 6 divided by this.
Yes.
Is not a protein. So it's still a process on the second time scale. I'm itching for something that goes, so I want to scale
I
that.
think like this divided by number of proteins would be the time required to generate one protein. So 10 to the power minus 3 seconds.
No, say that again.
3000?
Yes.
10 to the power 6 protein in 3000 seconds.
Yes. Yeah.
One protein in
Yes.
3000 by 10 to the power 6 seconds. It's an average thing. But
It's an average, yeah, sure. But, okay, so then we're going to milliseconds.
yeah.
Okay,
Yeah.
fine, good. So now we've gone to one's orders of magnitude faster, and now we're getting into the regime of how long does it take to take a protein like this that looks like a spaghetti chain and fold it, give it some secondary structure, and we're going to get into this more deeply, okay? But I have a protein that's generated by the ribosome. It is a polymer. It has a structure, unfolded structure. This may not be its thermodynamically favorable state, and so typically what will happen is, in order for it to function, it's going to have to transform from this to something that looks folded. So all these examples, let me go back a bit to something I showed you earlier on. Here we go. All these examples were proteins in their folded conformation. So they have some. So how many of you play Lego or used to play Lego? No? Nobody plays Lego?
We used to play with dirt.
You what?
Dirt.
Dirt.
When we were kids.
Okay. I see. Okay. Okay, so I still play Lego. Okay? And you know what Lego is? Lego. I know, okay, the boys know Lego, but the girls don't know Lego. Lego is, it's the toy that allows you to build objects. So you start with from small pieces and you put them together and you can make buildings, you can make machines, whatever. So these polymers, these proteins, are made up of Lego pieces, okay? And when the Lego pieces are not in a particular form, they can't do their function. They can't have a biological function. They have to change into some specific shape. Only when they change to a specific shape will they be able to perform their function. It's just like if I have all the Lego pieces sitting around without a particular structure, it doesn't do its job. Okay, so something has to go to get the proteins from this to this, and this type of time scale is milliseconds. Okay, it can be faster. Okay, but it can be milli to microsecond. Okay, so that's another time scale.
I expected it to be smaller than millisecond because millisecond we only
Yeah.
got for one folded protein. So we can still divide it by the total number of stages.
Yes, sure. So there's some that fold. So the range of time scale is micro to milli. Okay, but there are some proteins that still do fold on a slower time scale. Okay. Okay. So I have seconds, milliseconds, microseconds. Well, we can still go further down.
I think if you go further down,
Yeah.
maybe we can focus on water.
Okay, let's do
because
that.
it has to form on a faster rate because we need 10 to the power 4 times more water. So just by dividing 3000 by 10 to the power 10, you will get a lot down.
Well,
So waters are like generated on a faster rate, so lesser time scale.
so, okay, that's, okay, hold on, let me go back.
Maybe if I may ask, is it generation or just the process through which water enters the cell?
No, but
Yeah, this is it. So this is a very good question. Is it the time it takes for to bring are water molecules really generated
Only just
in the cell, or is it the chemical entity exists and it's being picked up? So this one would be waters in the bath outside and how fast you get them in. Then of course, generation of water, we're talking about different energy scales.
yeah.
And so bonds, covalent bonds being formed, being broken. And so there, we're talking about another story, which we'll get to. Okay, so that's catalysis, enzyme catalysis. Those are actual chemical reactions. So here, I'm not talking about chemical reactions. Okay? But focusing on a water molecule or focusing on a covalent bond, so we already talked about this in the numerical class, the typical frequencies associated with those types of things are very large. So we're talking about greater than 3,000 wave numbers, high frequency modes, things that oscillate on the what time scale? 1 what? 1 femtosecond. Okay, those are very fast time scales. Are those relevant to biology? So, yes, they are, especially when you're dealing with excited state processes, optical processes. Okay? So, remember I gave you the example of vision. So your eye is able to view objects because photons interacting with specific proteins in your retina optically excite the molecule. After they're optically excited, there's a change in the secondary structure, the conformation of a protein, and this triggers out a signaling pathway, which is then interpreted by the brain. Those initial steps are going to be on the femtosecond time scale. So very, very fast time scales. Okay, so this we already discussed. Yeah, please.
What about like you put your hand on the fire?
Yes. Why would you do that? But I don't know.
I mean, before you put your hand, you are removing because of the pain you feel. So, and I know that from the biology perspective,
Yeah.
the process is a very lengthy process.
Yes.
So how long does it take
Well,
for you to realize that it's painful?
sub-second, right? I mean, and it's, so what, I don't know if we're going to get to this today. And part of the reason for those fast response times is because signaling along neuron cells involves diffusion or motion of proteins on a much faster time scale than you would expect from a Brownian random walk. So if you assume signaling being caused by proteins involving a Brownian random walk, it would take many years before you would remove your hand from the fire, and then you'd be dead probably. Okay, so and probably the way we evolved over time was, response to stimuli. Lots of people burnt their hands when they were discovering fire and whatnot. But I mean, we're talking about, there's a hierarchy of time scales. There's a hierarchy of time scales. On the sub-second time scale, your brain already induces a mechanical response from the muscle proteins in your body to withdraw your hand. Okay, but we're going to get to your question in more
I
detail.
just wanted to
Yeah,
add one, like, a little bit out of track comment.
sure.
Like, especially in this kind of experiment,
Yeah.
about response and all this
Mm.
stuff, it may also be more of a psychology than just biology, because it's the placebo is in our mind. So maybe, okay, it burns. So,
What's the difference between psychology and biology?
for example, if someone doesn't know what fire is, like we do this experiment,
Yes.
then would his response be a little bit later than the one who knows, okay, what fire does and all these things? Because it's already in the mind. Okay, it hurts.
Yeah, but so
Like,
psychology makes you
like bias.
not, well,
Like bias.
sure, but psychology makes you not want to bring your hand in the first place. You're resistant to it. Okay, but let's say I took two people, and I'm the one who forced their hand to the fire. Okay, I would get in a lot of trouble for doing that. But let's say I did it. I think if you then were able to measure the firing of the pain receptors, which you could do, you would, I think, find that basically the response time is the same. So I can't see, I don't understand why you think the response times would be at the molecular scale.
But okay, so let's suppose that there is a certain group of, let's say, deer
Yeah,
in a certain national park in a certain area,
yeah.
and initially they don't have any predators.
Mm-hmm.
So, okay, they are fine for years and years.
Yes.
And let's suppose that they die in a separate way.
Yes.
If they don't, like, the system will grow.
Yes,
But then we introduce a pack of wolves,
yes,
and after that they start developing response.
yes.
So, do you think introducing the wolves and letting them grow for a few generations will change their biology, because now they have
Okay. So if you pose the question like this, then I will say yes. So let's say the neural networks that are created will evolve at a biological level to optimize response. This I will agree with, yes. But yeah, I guess my problem was,
on this one.
yeah, no, that's, let's say, it's not clear to me that one can distinguish between psychology and biology. To me, they're the same thing. But, yeah,
For me, I think the best thing is to close maybe Joy and Nishan, cover their eyes and introduce a fire between them. Then you think.
okay, I have a suggestion. Why don't you guys do this after you graduate in August?
No, I think the problem would be like an experimental problem. Like, we really won't be able to calculate the time. Like, we don't have a clock to so that.
No, you.
In our
Yeah. This is clear. This is clear. Okay, good. So, these guys, the proteins, tumble. They're moving around. Water molecules are moving around. They're breaking and forming. And the time scales associated with the breakage and formation of hydrogen bonds of water, the time scales associated with the tumbling or fluctuations of a protein are on the order of picosecond to nanosecond. Okay, so this is, now we've gone from femto to pico and nano. Okay? Okay. Yeah, please.
There is something that I'm curious about.
Yes?
How do proteins produce other proteins? I thought that when I eat proteins, then that's when I can have proteins. If I don't.
Well, so there's something called your stomach. Your stomach digests protein. When it digests proteins, it breaks it down into amino acids. Okay, so and then those things are used in various other biochemical networks. Your proteins are encoded, come from DNA. Okay? In order for the DNA to replicate and to work efficiently and to convert things into RNA, for RNA to be converted into protein in the ribosome, you need other proteins. Okay? And those other proteins are involved in helping facilitate many of these biochemical pathways. But you know, it's not that you eat the meat protein and those protein in the meat go and do function. It doesn't work like that. Okay, that's used to typically to get energy. Okay, and then used as building blocks for other things. This will make much more sense when we get into the further lectures. Okay, so I already said this to you. Protein folding is this problem. So this is one of the standard problems that sort of interest to physicists. I have this spaghetti chain. How does the protein find its native conformation? Okay, that's the protein folding problem. And so people spend a lot of time trying to understand the free energy pathways, the role of entropy and enthalpy in driving protein folding. But we're going to get into this. What I want you to keep in mind for now is the time scales. Okay? And basically this just summarizes all the different time scales in biology. So we're going from all the way femtosecond to, seconds, minutes, hours. Okay? So this is what makes biological problems sort of very interesting and also very challenging. It's because you've got a hierarchy of length and time scale that you have to deal with. So for people trying to develop models to understand biological problems, you're stuck with this challenge, is having to deal with things on lots of different time scales. And the point is that you may say, well, there's a separation of time scales. Not really. So there are processes that can happen on the nanosecond time scale or the microsecond time scale that can affect something that's supposed to happen on this time scale. So imagine this process happening. It's on a microsecond time scale, and there's some chemical reaction that involves this protein. The exact details of the architecture of the protein may sensitively affect something that was supposed to happen on this time scale. Okay, so this makes them very interesting, but also very challenging to work with. That's the first point. Second point is, well, the reason why there's a big hierarchy of timescales is that they're soft matter systems. Okay, and that's why you have so they're highly enharmonic. Okay, highly enharmonic, highly corrugated, so very glassy, like rough, free energy landscapes. Okay. Okay. So we've basically sort of alluded to this already, but I'll just complete the conversation. So I'm not going to get into this right now, and actually I'm the wrong person to talk to you about this because I don't even understand it myself, but it's not important. So there's a lot of complicated biology behind the cell cycle. Okay, for us, What we care about is I need to go from 1 cell to 2 cells, period. And this takes about 3,000 seconds. Okay? And so what are the implications on this? Well, one is that in order to generate 2 cells, I need construction material. I need the objects, the Lego pieces to build my protein, to build my cell membrane, to build my DNA. And more or less, using the estimates that we used before, just focusing on carbon, because that's one of the most abundant elements in organic matter. There are about 10 to the 10 carbon atoms needed to generate a new cell. Okay? So you can sort of see this. You take 10 to the 6 proteins and you multiply by some factor corresponding to the number of carbon atoms roughly in a single protein. Okay? So it's on the order of 10 to the 10 carbon atoms. So let's say that the carbon atoms come from glucose, sugar. Okay? Well, if I need 10 to the 10 carbon atoms and it takes 3,000 seconds, basically to generate a new cell, I need to absorb about 10 to the 5th glucose molecules per second to generate a new cell. That's, I mean, it's highly non-trivial. And okay, this factor of 6 comes because there's 6 carbon atoms in glucose. Now, Nishan already answered this for us at the beginning of the class. Number of proteins that I need to absorb to generate a new cell, 1,000 proteins per second, and 10 to the 6 water molecules per second. Okay. You know, that's very, very, very efficient. So what is it that allows for such type of efficiency? If I just assume that water molecules were undergoing a Brown and random walk, would this be the timescales that you could achieve to generate a new cell? Question?
You talked about 10 to the 10 carbon atoms.
Yes.
Okay, I'm not so sure about the mass of one carbon atom.
12 atomic units.
12 ATU. Okay,
AMU, yeah. Atomic mass units.
Which is
12 into 10 to the power minus 27 kilograms.
But okay, that was just your side question.
No, I just thought that there is this mass they always give in grams for any element. I don't know, is it?
Yeah, that is
Yeah.
the molar mass.
That's a molar mass.
Oh,
Yeah.
so the whole
So it has got 12 grams in 6 into 10 to the power 23 carbon atoms. It has got 12 grams.
So I was trying to multiply 10 to the 10 times that, and I was like, would that exceed the mass of the cell? Now it's okay.
Yeah, there's molar mass and then mass. Okay. So, you know, so at least when I remember when I saw these numbers, I was quite amazed and surprised at how efficient the biological machinery works. And so what we want to do is to understand what are the physical driving forces that allow for this type of efficiency. Okay, that's what we're going to try and build our intuition on. So I don't know. Let's give this example just since we've talked about it. Okay, so Brownian random walk. The time scales associated with the diffusion go as x squared over d. Okay, so the diffusion constant goes as L squared over tau. Okay? Now, if I take my E. coli cell and I want to know how long it's going to take for a protein to diffuse all the way across the E. coli cell, then I can calculate that. I can get an estimate of this. I know the length. Let's say it's a one-dimensional thing. D is the diffusion constant. I can estimate the diffusion constant by using this Debye-Stokes hydrodynamic theory, which relates the timescales of diffusion to thermal energy, the viscosity, and essentially the size of my object. Okay? So if I plug in a typical protein, radius 25 angstroms, choose the viscosity of water, let's assume it's in the bulk, I will get something on the order of 100 micrometers squared per second. So this says that the time it takes for this protein to diffuse across to the other end of the E. coli is 1/100th of a second. Okay. Assuming that it's undergoing a Brownian random walk. Okay, so let's keep that in mind and actually go to a question that you were, I'm actually happy that you asked that question because it motivates what I'm going to show you. Let's consider a neuron cell, okay? Neuron cells can be very, very long. Okay, they can extend all the way from your foot. I mean, they're very long, okay? So let's imagine this is the axon of the neuron cell, and let's imagine a protein hopping across the neuron cell using the time scale that I invoked from the previous slide. It would take 3 years for that protein to move from to travel 10 centimeters if it was doing a Brownian random walk. Not going to happen. You're going to be dead. All of us will be dead. So something else is going on. There's some underlying potential. There's some directionality. There's something avoiding the protein from doing a random search. Because if it did a random walk, it would basically take very long, or the age of the universe, for protein folding to happen, for diffusion, important for signaling to happen. Okay? And so, what drives, what controls this directed motion? What is it that avoids noise in the environment from, making the diffusive or the dynamics random? So that's what we want to try and address. Okay. So I'm going to skip this because this is going to come later, and I want to move into this question of why the nanoscale is special. Okay, so before we do that, let's start by getting a sense of some energies. So, covalent bonds. How strong are they? So, yeah, how much energy do I need to break a covalent bond? Like a carbon-carbon bond. I'm going to put this as 2, I'll put this as 3. The first one, let's put thermal energy. That's the baseline. We need to always fix thermal energy. Now, with respect to thermal energy,
You are looking like in terms of a rough figure or
give me a rough or a precise one, whatever you want.
I can't.
Just take a wild guess.
Maybe I should just let's demo which one is stronger to work and which one is better to work.
So wait.
Say maybe I should just
Okay, fine. You want to make this strong and weak.
Yeah, I'm just classifying them of weak, strong.
Fine. If that's what you want to do, do it. Now tell me.
Okay, then it should be so thermal. Hydrogen is a weak gas. I mean, it's easy to work it, very easy. Then
Wait. First, is thermal energy, does it depend on the length?
no.
No? Okay, it's fixed by?
Like when you give it KBT.
Okay. So it depends on the temperature. Okay, so I fix that. I fix KBT to, for the sake of simplicity here, let's keep it to room temperature. Okay? Let's do that. So room temperature. 295 Kelvin. Okay. So now, relative to this, where's this energy to break it? How much energy do I need to pump into the system? So do you think in terms of EV or in terms of another energy unit? You think in terms of EV. So tell me how many EV? Two EV?
Not two. Maybe approximately two milli EV.
One EV.
No, not one. Approximately in terms of milli.
So okay. Milli, are we like at milli scale, milli EV or
Milli EV.
you say milli EV? Who agrees with that? Voila, you agree with that? You think? William, your thoughts?
KBT is about 25 milli.
2 kBT.
Yes. So in terms of
2 kBT. If your covalent bonds were 2 kBT, let's say, if it took 2 kBT to break them, basically we would all be soup right now. Because basically I'm coupled to a bath right now that's keeping, we would all be soup. In fact, we would be fried soup because it's not only we're not at room temperature, we're at 37 degrees Celsius, so we would just dissolve. So let me put, so this, I kid you not, so I've said this to you before, if you, yeah.
if I say two, what I'm simply saying is like 2 times what you have in the thermal energy. So it's approximately
but-
50 electron volt.
That's still small. It's very small. I mean, if you just think about the magnitude of fluctuations in thermal energy, or energy at room temperature. Thermal fluctuations alone will cover several tens, on the order of 10 kBT, 10 times that. So it's too. Let's back up a second. In a covalent bond, what makes up a covalent bond?
The number of pressure.
Okay, so it's the sharing of electrons. Okay.
So
And that, yeah.
so if you have so many slash brackets, so many pair sharing, one, it takes a lot of. So what I'm saying is approximately for one batch.
Yes.
So if you have so many of them, then you have to increase the energy as well, because you have to break one by one.
But I think that will scale linearly with the number of sharing.
Yes, that's what.
But that doesn't affect much.
Doesn't affect much, yeah.
But we have to focus on per broken.
Exactly.
So hydrogen, we know that it's 13.6 electron volt.
That's the ionization potential. That's all right.
To
Yeah.
knock it out.
That's
So we are
Okay.
expecting a lesser,
exactly.
but in electron volt, maybe somewhere, I don't know. We just need to dissociate the electrons among each other.
Exactly. You have to
Yeah.
split them apart.
Yeah, and also like for carbon, it has got like many orbitals before it's screened.
Yeah.
So it's definitely lesser than hydrogen in first place to
That's-
ionize.
That's
Or maybe there are more nuclear, but okay, let's cancel them each other.
Yeah.
So let's say it's the same. So it will definitely be lesser.
okay.
I don't know, maybe 20.
A fraction.
Yeah, fraction. Yeah.
Certainly some.
I don't know. Like,
So order of magnitude, of course, depending on the type of atom, the screening, several eV. This is the order of magnitude. So several on the order of 100 kBT.
Yeah.
Okay, that's order of magnitude. This will change depending on a single bond, double bond, triple bond, whatever. But we can certainly say for sure that there's a separation of energy scale of breaking that covalent bond versus, let's say, weaker interactions that don't involve electron sharing. So the moment I have electron delocalization, the moment I have electron sharing, I'm going into a regime of much more energy being needed to break apart the bonds. Okay? Now, this is something that you have to keep in mind. I'm just giving you a tip for your interviews. If you plan to CISA, these are the types of things that they'll ask you very simple questions, and they want to get a sense of because more complicated things, well, you can always look up in a textbook, but you have to have a feeling of these types of things when you're doing your research. Because then if I compute an energy from my model, and I say that the energy to break a covalent bond is kBT, then I know something is not working. So I have to have a good sense of these energy scales. Okay, so here we say hundreds kBT. Okay? Oh no, you're still not convinced. It's too much. Well, so I have two responses to that. One is that it's a fact. So this is something that is known experimentally. Okay? And two, you have to, sort of electron movement. So when you're moving an electron charge, and there are other electrons around, or a proton charge, what the energy scales that you're going to be working at are,
It's hydrogen.
it's
The
EV.
upper limit is hydrogen atoms, ionization energy. So
It has
to be in that. It
It has to be in that
great.
regime. It can't be less than that. It says this is just a consequence of that. Okay, covalent bonds, hydrogen bonds. Okay, these are, I should say, electrostatic, plus screening. So, classical electrostatics. Okay, this roughly 10 kBt. So, if you want to think of it as strong and weak, fine. But there is a separation of energy scales. Okay. Now, why are we discussing this? Well, several things. My biological system is at room temperature, or not room temperature. My biological system is fluctuating in response to thermal fluctuations, thermal energy. In my biological system, there are processes involving formation, breakage of covalent bonds, and there are biological processes involving the breakage and formation of electrostatic interactions, screened and unscreened by the dielectric medium. So I have to figure out and think how thermal fluctuations are going to be able to drive processes involving all these different energy scales. That's where we're going. And give me a second. What I'm going to try and show you is that it is only at the length scale of nanometers, only at the length scale of nanometers, where there's a convergence of different forms of energy: mechanical energy, electronic, electrostatic. Okay? It's at no other length scale do these energies converge. And at this length scale, thermal fluctuations can allow for efficient conversion from one form of energy to the other. Yeah?
I'm inquiring whether it's important to talk of some fluctuations, yet we know that most of the living organisms are endothermic. They can maintain temperatures.
No, so wait. You're talking about a very different concept here. So every system made up of particles in contact with a heat bath in the canonical ensemble undergoes thermal fluctuations. So, okay, I'm not going to get distracted into this conversation now. We want to talk about it, let's talk about it after class, okay? Okay, so let's keep those things in mind. So there are many, many different types of forces, interactions, forms of energy that we need to think about in terms of in biological systems. For example, if I want to bend a DNA molecule, if I want to stretch a protein, I'm going to be doing some elastic type of energy. If I want to break hydrogen bonds, or I want to bring charged particles in a liquid environment close to each other, I'm going to be doing some electrostatic work to bring those particles together. Okay. And so we're going to try and consider and think about how these different forms of energy are efficiently converted from one form to the other at finite temperature. So at room temperature. Okay, so we've already talked about this, so this just you have it here for your notes. So very large numbers, very small numbers, well, very small numbers, on the order of 5 to 10 kBt more or less. Okay, so now let's come back to covalent bond. So we just had a discussion that the covalent bond involves electrons being shared in some confined region, essentially. So if I wanted to know what would be the simplest physical model that would allow me to understand what energy required to confine an electron to a specific region is. So I have an electron, okay, I want to confine it in a small space or a big space. What would be the simplest model that I could use to understand how the energy changes as a function of the length, the confining length?
Horizon, yes.
Too complicated. I want something simpler. Particle in the box. So I want my particle as the electron, and it has a localization or delocalization length. And if I want to just see what the effective binding energy of, or the energy associated with confining the electron to a specific length is, I would use a particle in a box. Okay, so I'll end with this, and then we'll continue on Tuesday. So for a particle in the box, so I'm looking at covalent bond confining the electron to a specific length. Okay, so how does it go?
Energy, 1 by L square. N square.
Say it again?
1 by L square.
Okay, then there's an H
N
bar,
square,
blah,
T
blah,
square.
blah. Okay. So I have got one formula that allows me to understand how energy scales as a function of length for the particle in a box. And this, so the length 20 minus 3 to 10 to the 10. And this is in joules. This goes to 1. This goes like this. And so I'm going to put (10 to the minus 9) there. This is in meters. This is the length. And thermal energy is, okay, this is, the scales are off, but I'll do this more cleanly on Tuesday, but I just wanted to get you to think about it. Thermal energy is somewhere here. It's 10 to the minus 21 joules. Okay? So this is our first data point. Thermal energy, independent of length, binding energy, or the energy required to confine the electron to a specific space, which we're using as a proxy for creating a covalent bond. And where we're going to go with this is the following. So this is one length scale dependence as a function of energy. We're going to do a similar thing for the elastic energy. So I can ask, what is the energy required to bend or stretch a rod of a particular length? And what we're going to discover is there's something very special about this region here. Okay, it's already, well, you can't see it from this, but we're going to see that it's only at this length scale where there's a convergence of thermal energy with other forms of energy. And that convergence allows for very efficient conversion of energy from chemical to mechanical, chemical to electronic. And this only happens at the nanoscale. And that's why biological systems work as efficiently as they do. Okay, so we'll stop here for today. I will start again on Tuesday from here. And don't forget to do your homework. See you at 2. Nika is sick, or she's around, or do you know? She's?
I think she's in the library.
Ah, okay, okay. Yes, please. So, yeah, so we're talking about macroscopic things versus microscopic. So,
Fluctuation is in temperature.
no, every system, okay, at room temperature, let's say your protein, okay, will undergo, so KBT is fixed,
I wake up in the morning, I do everything
okay,
fast.
but let's say your object will exchange heat and energy, right?